Hello, and welcome to Let's Meet the Virologists, a podcast about the people behind today's virology headlines. People just like you working to understand viruses and how they affect you. During the COVID-19 pandemic, we are talking with faculty involved in coronavirus and COVID-19 related research so that you can learn who they are and what they do. I am Larissa Thackeray, and I am hosting this podcast from America's Heartland in St. Louis, Missouri. 2 RNA vaccines for SARS-CoV-2 have received emergency use authorization by the FDA, with vaccinations with one of them taking place for workers in healthcare settings this past week. On December 17, 2020, we talked with Dr. Rachel Presti, an associate professor of infectious disease and director of the Infectious Disease Clinical Research Unit at Washington University School of Medicine. Rachel received her MD PhD at Washington University studying murine cytomegalovirus. She did her residency in internal medicine and a fellowship in infectious disease. She also did a postdoc with Skip Virgin identifying new herpes viruses. She has run clinical trials for HIV, HCV, influenza, and now SARS-CoV-2. Hi, Rachel. I'm happy to have you with us today. Why don't you tell us about yourself? How did you become interested in virology and medical research?
So I sort of always was. My father did data science for Boeing, and as part of that, he did a lot of computer science, and they had access to huge supercomputers back in the 70s and 80s. And as I was growing up, he would talk to people from all different sort of science areas and come back and say, you should be a meteorologist, or you should be a microbiologist. And so it was always sort of a science thing. So I guess that was sort of the start, was that he always encouraged me to do something with science, and I was always very interested. And then in college, I was a biochemistry major, and just really always liked microbiology and virology. I guess I spent quite a bit of time in that, although I did a lot of chemistry as well. And then I came to WashU in 1994 to do the MD PhD program, and sort of always thought I would be more of a lab person. I worked with Skip Virgin and did work on latent cytomegalovirus and the virology of that. Although I guess to some extent that was one of the really exciting things, was toward the end of that, we had some data that suggested that interferons blocked reactivation from latency, and there was some interest in the pharmaceutical area in maybe looking at whether or not that could be protective in transplant patients, or and so it was kind of interesting even then to think about the sort of translational impact of your findings. And so I have kind of a winding pathway. So I went back to medical school and finished medical school and did infectious disease. So I wasn't sure I wanted to do medicine even, but if I was going to do it, I was going to do infectious disease, because I found that incredibly fascinating, and always felt like the one nice thing about infectious disease is it's a foreign organism that is causing an infection. So there's always the hope that you can treat that in a way that's specific to the organism and not hurt the person. And so unlike most of other medicine, it always felt like there's always a possibility of treating or preventing infectious diseases. And so you could help people. And that's what I really liked about infectious disease. It's also nice because for somebody who likes to do a whole variety of things, infectious disease is always changing, and you always get something new. Maybe you don't always need something as new as SARS-CoV-2 in my life, but it's always interesting. You always have a new issue. You always have a new problem to solve, and then we also always have new therapies that are being developed. So new antibiotics, new vaccines, new treatments, new strategies for dealing with infectious diseases. So I liked that a lot. I did go back into the lab after fellowship And what I did find eventually was I started looking for new viruses again with skip virgin. And what I liked about looking for new viruses was the patient interaction and the interaction with clinicians who were trying to identify the reason that somebody had some clinical syndrome, and had collected samples, and then we looked for new viruses. And the lab pathway was to study the viruses, and what I liked was studying the people. So that was about when I changed course again and really went towards clinical research, where you're actually interacting with people all the time. And then luckily I've had the opportunity to transfer that into also much more translational research, not just straight clinical trials.
To follow up on that, can you describe a little bit of the work you've been doing after your postdoc and before COVID hit in March?
So we've had a clinical trials unit that was predominantly an AIDS clinical trials group since '87 at Washington University. And so I started working with them, and I started out doing clinical trials with HIV. And so treatments for people who developed, had resistance to a lot of the drugs. We got access to some of the newer agents, and were able to treat people. And that was really, really very satisfying to be able to take somebody who couldn't get undetectable and be able to give them a new drug and have it be so powerful that that even if they didn't take it exactly the way they were supposed to, they could get undetectable. So as part of that, about that time, they started developing new therapies for hepatitis. And since so many of our HIV infected patients were co-infected with hepatitis B or hepatitis C, I took over doing a lot of the clinical trials for hepatitis C. And that was incredibly fun, and moved really, really fast. It was almost like HIV on fast forward. So instead of, decades of research to get to therapies, we had, a couple of years before we had new therapies for hepatitis C that were much safer, much better tolerated, and much more effective than interferon, which is what we'd been using for a while. So I started doing that, and continued to do the HIV work. And then I always had a little bit of an interest in maintaining some translational work. And so we did some work looking at the microbiome with folks from Skip's lab. So Scott Handley was one of the main ones I worked with, to look at how the microbiome changed and how that might have impacted on some of the pathology of HIV infection. So started doing some microbiome work, and looking again at sort of immune responses and inflammation in people who are HIV infected. I guess we've managed to transition as well. So continuing to do a lot of clinical trials work, looking at new treatments, but moving into the translational area. So looking at obtaining clinical samples that can be evaluated in the research lab at a much more detailed and higher level than you would ever sort of evaluate them on a clinical scale, and see if we can figure out why do people have certain phenotypes? Why do people not have good immune function restored? Why do they have higher levels of inflammation, and so on? And then we started working more recently with Ali Elabidi to look even more in the translational realm at how influenza vaccine works. And we're able to do that in a way where we could enroll people who hadn't recently gotten influenza vaccine to get influenza vaccine, and then get even more detailed clinical samples, so not just blood, but to get lymph nodes and bone marrow and really understand what is the immunology and the long-term immunology of response to influenza vaccine in order to try to improve that. And so because of that, we were positioned pretty well then when COVID came around to do the whole gamut of things from basic cohort analysis to collecting samples for translational and basic science work, to trying to understand now how the COVID vaccines work, using similar methods that we're using with Ali.
Right. So to follow up on that, can you tell us in a little bit more detail some of the different COVID-19 related research that you've been doing?
Sure. So the very first thing we did when it became apparent that that SARS-CoV-2 would actually become an infection that we're seeing quite a bit of in the U.S. So back in March was to establish some cohorts of people who were presenting to the ER with symptoms concerning for COVID, enroll them, and then collect longitudinal samples from people, both who tested positive and some who tested negative as well. So comparative samples. So we wound up, I think, with 500 people enrolled to that cohort. Not everybody has maintained long-term follow-up, but we have samples then from long-term follow-up from those folks. A similar cohort was established then because of work we were doing with Jeff Henderson to try to get convalescent plasma up and running as a therapy. Part of that, when it was initially being developed, was the need to let the blood banks know who had cleared infection, so when they needed documented infection and documented clearance for safety purposes, and then had detectable antibodies. And we've enrolled a couple hundred folks in that cohort. Once the blood banks were able to take over that analysis on their own, we continued to follow people as a longitudinal cohort, looking at antibody responses and then collecting information about long-term symptoms and collecting blood and saliva for banking so that people can look at those sort of longitudinal samples. So those are the first things we did. We also very quickly got involved through Steve Miller with Express Scripts in a study at the time. The treatment that everyone thought would work was hydroxychloroquine or chloroquine, maybe with azithromycin. And we designed a study with Jane O'Halloran and myself to compare those therapies. So there were randomized control trials going on already, and what we wanted to do was look at a sort of treatment strategy study. So we had 4 different arms: hydroxychloroquine or chloroquine, with or without azithromycin, and enrolled people to that study until the data started coming out that it really wasn't. None of those were particularly effective. You know, unfortunately, a lot of that was hurt, I think, by sort of how political things got, and so it was hard to enroll people as well. So about the time that was going on was when we first heard that there would be vaccine studies. They were initially supposed to start in July. I think they actually did wind up starting more in August. And through the work we were doing with the AIDS clinical trials groups, those are NIH-funded networks, and they established a coronavirus vaccine prevention network that was a group of HIV clinical trials and vaccine clinical trials networks that the NIH was already supporting. So we got involved in that. We're working with St. Louis University as well as another site for those, and started getting ready to be able to do the huge phase 3 clinical trials for COVID vaccines.
Great. And did you also do a trial for any kind of monoclonal antibody therapy, or no?
Oh, yeah, sorry, we did that too. So we did. We've done a couple of different trials then. We've done trials with monoclonal antibodies both inpatient and outpatient. And so the outpatient clinical trials, we're looking at the Lilly antibody. The inpatient clinical trials, we're looking at the Regeneron antibody. And so we did those for several months. We actually are still part of an adaptive outpatient clinical trial that did the Lilly antibody and is now starting to think about pulling in other antibodies or other therapeutics that could be used in the outpatient setting. So we're on a bit of a pause on that, but are expecting to have new agents to test in the future. So part of the bamlanimab approval for EUA was some of the data that was collected as part of those studies. So we're sort of eagerly looking for new, easier to administer outpatient treatments for COVID. I guess the other thing that we've been involved in is an inpatient active-1 study to look at different immunomodulatory agents in combination with remdesivir to treat people with more severe COVID requiring oxygen and requiring hospitalization.
Okay. Wow, that's a lot of trials.
Yeah. We also did the expanded access programs for convalescent plasma and remdesivir starting in April, May, to get those to the point where they were available on emergency use authorization.
Right. So I guess to take a step back, more generally, what has been the most exciting moment in your career so far?
It's sort of more like little moments. I talked before about starting clinical trials and getting to give people a new protease inhibitor called darunavir. And I remember we had a participant, and I was, she just did not like taking medications, and this was a twice-a-day medication, and she would come back, and I'd realize, oh my gosh, you've only taken half of your pills, and you've returned half of your pills. And so when her viral load came back, it wasn't undetectable, but it was less than 100, and I thought, this could really work. Like, I could really help people, and this is going to really help people who have difficulty taking medications. So there's things like that. There's, every time you get a clinical trial result that is able to change clinical care, I think that's always exciting. I mean, it's hard because it's not like that aha moment that you talk about in the lab where you think, this is where I knew that I had the answer. It's more of a slog, but when you actually realize that all that work you did on, study X is going to result in actually better treatment for somebody, that was just really exciting. So getting to work on the new hepatitis drugs was fun. We've been involved with long-acting injectable drugs for HIV for treatment and prevention. And, anytime you get something that is superior to the current standard of care, you feel like this is helping people. And so it's like little things like that make it, just really, really fun. So.
And I guess conversely, what is the most difficult thing you've had to overcome as a scientist or as a doctor, and how did you overcome it?
So I think the hardest thing for me was the decision to stop working in the lab. So I loved working in the lab, and I had a lot of fun. I really enjoyed it. I liked what I was doing. I felt like it was important. It all sort of happened around the same time that I had kids, and I guess what it came down to was I was getting more satisfaction out of the clinical work that I was doing than the lab work. And but it was just really, really hard to give up that idea that I would be, the triple threat with the basic science lab and the clinical work and the teaching, although I was never great at teaching. And so what I think what really sort of helped me make that decision was both the support of my division chiefs, and just thinking like, what is it that is worthwhile leaving these like new children that I have that I absolutely adore? And you know, what is it? What from a professional standpoint is important enough that you need to do that and not spend all this time with these people that these little people who you've fallen in love with.
And I guess to follow up on that, if you had a chance to ask your older self, say you 70-year-old, you know, getting closer to retirement, one question, what would it be?
How long are you still productive? How long do you stay doing this, and still feel like you're contributing? So, you know, I guess I would want to know, you know, some of this is really hard and really tiring and really stressful, because I feel very responsible for all my clinical trials participants, for my patients, and what I'd want really would be confirmation that at the end it was worth all the worry, that I actually did make a difference, but I felt like I made a difference in someone's life.
And then I guess turning more back to the COVID-19 pandemic, how has it affected you as an individual?
Oh, I've never been more busy in my whole entire life. I've never felt more responsible for not just my own little, my own little world became something that was really important to like a huge part of the population, I guess. And so I felt, you know, early on in the pandemic was incredibly difficult, because I felt like I needed to find the studies. I needed to be able to bring treatments to people, because, you know, we didn't know how to treat it. We didn't know what to do. And then when the vaccines became available, the vaccine studies, you know, we just really wanted to make sure that we were enrolling the right people, that we were getting the word out, that there was enough information coming out, that people knew what we were doing and that they would trust it at the end of the day. So I guess what changed the most for me was I was continuing to do a lot of clinical trials, but I have done a whole lot more actual outreach into the community, and so media, and talking with community groups, and not just doing the research, which is what I'm used to doing and feel very comfortable with, but actually figuring out how to communicate that to people so that they understand what we're doing, so that when we do have treatments or vaccines, that people feel like they were done well, and they were done by people who cared and were honest, and really had their best interests at heart.
So have you managed to find any free time during the pandemic? Have you picked up any new hobbies, or have you just been working?
I've mainly been working, but the one thing that I did start doing was really long walks. And so initially that was a stress release, and now it's just a habit. But I think I know every house in our neighborhood. I've seen them in all seasons. I think there was a health initiative that happened right in March, about the same time that COVID really became real, where they were encouraging people to do 10,000 steps a day. And so I thought, I can do that. And so I went through that, and then I just kept on going. And so that's the one new thing that I've done. Things changed a lot. My husband used to travel a lot more, and obviously that all went away, and he was working from home, and the kids were doing school from home. And instead of it kind of got simpler to some extent. I mean, we were working a lot, but so much of that, driving around and feeling like you're just being a crazy person when you have teenage kids, taking them here and there and different places and trying to make sure that, that everyone's home at time, and can have dinner together. All that went away because they were all always home. And so it became easier. But yeah, walking is sort of the one thing that I've started doing. I don't know. The kids play the piano and the violin, and I play the piano too. So we try to do that together some too.
So I guess to follow up on that, sort of as a virologist and as a doctor, how do you make decisions about how to keep yourself, your family, and your community safe from COVID? How do you sort of think about the risks involved with doing different things?
I try to listen to the epidemiologists. I try to listen to the folks who do a lot of occupational health and infection prevention, and try to really think about what the science is and recognize that we're not always right initially, and that things might change and you might have to change your practices when we have better information. So initially, we weren't wearing masks a whole lot because that wasn't the recommendation. And as soon as it became clear that that was actually going to help, we moved to doing that, making sure that the kids had their masks and that they were doing what they needed to do when they were out in either in school or both of them do athletics. So making sure they always had a mask that fit and they were comfortable with. So yeah, I mean, I try to keep up with what the guidance is. I think initially I was very obsessive about that, and then realized that was making me crazy. And so, it's nice being in the infectious disease division because you get all the advice, and you have people in your division who know more about certain things, and they're available and accessible to give you the answers of what you need. So I try not to worry about it. I try to do the things I can do. I can try to do the things that I can control. Wash my hands, wear a mask. We haven't traveled anywhere. We try to avoid gatherings. We did a lot of takeout and still do, and figure out what feels safe and feels right and balanced to us.
Great. Well, I guess we're wrapping up. Any last messages for our listeners? Any thoughts about sort of the vaccine, thinking about how that might end the COVID-19 pandemic? What do you want to tell people?
I want to tell people that they still need to wear masks and they still need to socially distance because our numbers are still appalling. We've sort of flattened out a little bit, but we flattened out at just a really high level, and we just can't handle, the hospitals can't handle another surge on top of what they're dealing with right now. I think the vaccines they're so exciting. It's really amazing and really a tribute to sort of like years and years of science that's been going on to be in a position where we could have vaccines that could be tested, that could be developed this quickly and could be tested this quickly. I firmly believe the data looks great and it looks like they work, but they aren't going to work till everyone gets them, and they're not going to work immediately. And so it could be a really, really, really rough couple of months until we get to the point where people are getting vaccinated and we have enough people who are immune that we can actually start to see the impact of that. So.
All right, Rachel. Well, thank you very much. Rachel's previous experience running clinical research trials for many other viruses allowed her to quickly pivot earlier this year to run a number of trials looking at the ability of different drugs, therapies, and vaccines to prevent or treat SARS-CoV-2 infection. These trials also serve as an important source of samples for translational research, trying to understand the immune correlates of SARS-CoV-2 protection and COVID pathogenesis. This has been Let's Meet the Virologist, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on most podcast platforms or at lmtv.podbean.com. If you are a virologist interested in sharing who you are and what you do, please contact us at letusmeetthevirologists@gmail.com.